It is a pleasure now to welcome Dr. Patience Odeniyide from Johns Hopkins Medicine. She did her MD at the University of Indiana and fellowship training at Johns Hopkins NIH combined program, and will be talking to us today on targeting oncogenic HRAS via vertical RAS pathway inhibition in HRAS-mutated human rhabdomyosarcoma. So, Dr. Odeniyide.
Thank you for the opportunity to share my research today. I'm going to be describing our efforts in developing targeted therapies for HRAS mutant rhabdomyosarcoma. So rhabdomyosarcoma is the most common soft tissue sarcoma of childhood, with sarcoma referring to malignant tumors of mesenchymal origin. It's the third most common solid tumor of childhood, with about 350 newly diagnosed cases each year. And as far as our outcomes for patients with rhabdomyosarcoma, those with localized disease have an approximately 70% event-free survival. Unfortunately, our patients who have metastatic disease or disease that have recurred after treatment have a much poorer 5-year event-free survival. So it's for these patients that we desperately need new and effective therapies. As we've heard previously, we broadly define rhabdomyosarcoma based on the presence or absence of a PAX3 or PAX7 FOXO1 fusion. So alveolar rhabdomyosarcoma or fusion positive RMS has the presence of this fusion protein, and embryonal rhabdomyosarcoma or fusion negative has the absence of this protein. And it's within the embryonal RMS category that we see mutations in a gene called RAS. It's present in about 22% of cases. RAS mutations are also enriched in patients with higher risk disease. So the focus of my research is on the embryonal subtype of rhabdomyosarcoma. So briefly on RAS mutations, the RAS gene encodes proteins that regulate signaling pathways in the cell that are needed for growth and survival. And when RAS is mutated, the cells then grow and divide uncontrollably, which ultimately leads to tumors. There are 3 isoforms of RAS: HRAS, NRAS, and KRAS, and we see mutations in these isoforms in about 1/3 of all cancers. Despite how common RAS mutations are seen in cancers, efforts to target RAS have been largely unsuccessful. There have been a variety of strategies that have been employed to try to target oncogenic RAS, including inhibition of downstream effector pathways, including the MEK-ERK pathway, direct inhibition of RAS using small molecule inhibitors, and then an indirect inhibition of RAS by targeting membrane localization. And so this strategy is the focus of my research. So the RAS protein requires membrane localization to signal downstream, and it undergoes a process called prenylation, in which the RAS protein acquires this lipid tail as a post-translational modification. This then allows the protein to anchor to the plasma membrane, and this is a necessary step for the function of RAS in the cell. The enzyme farnesyltransferase catalyzes this reaction for the HRAS, NRAS, and KRAS isoforms. And so this enzyme became an attractive target for drugs to try to inhibit oncogenic RAS. So Tipifarnib is an oral drug that is a competitive and selective inhibitor of farnesyltransferase, and it was developed in the 1990s and tested in a variety of adult and pediatric trials. And through these trials, we were able to see that Tipifarnib had a manageable side effect profile. But the outcomes that we saw for patients that were treated were modest, and so this was disappointing for the patients that received Tipifarnib. And so we think that this is likely because HRAS is the only isoform that relies on farnesyltransferase for prenylation. KRAS and NRAS proteins can alternatively undergo prenylation using the enzyme transferase, and thus can escape inhibition with drugs such as Tipifarnib that target farnesyltransferase. And so prior studies did not enroll patients based on the presence of an HRAS mutation in their tumors. And so there were likely patients with NRAS and KRAS mutations that received this drug, and that was likely why they were not sensitive to Tipifarnib. And so around the time that we were starting this research, many groups were investigating whether Tipifarnib might prove to be more effective in HRAS mutant models. And so this is data in head and neck cancers, in which here on the left, mouse models of HRAS mutant head and neck cancers received Tipifarnib, and we can see those with HRAS mutations had a much more favorable response with shrinkage of their tumors. On the right, this is a waterfall plot from patients who are heavily pretreated with head and neck cancers and who received Tipifarnib, and they were able to demonstrate objective responses in these patients. So this led our group to hypothesize that HRAS mutant fusion-negative rhabdomyosarcoma may be sensitive to farnesyltransferase inhibition with Tipifarnib. So I'm going to present a subset of data that we've recently published to address this hypothesis. We first wanted to demonstrate that in our models, Tipifarnib decreases HRAS plasma membrane localization. So we performed subcellular fractionation assays where we separated out proteins that were found in the cytoplasm versus those that were membrane bound. And here I'm showing a Western blot where the bands demonstrate the relative abundance of the protein. And we were able to see that when we gave Tipifarnib, we saw an increase in the cytosolic fraction of HRAS and a decrease in the membrane-bound portion of HRAS. And below, here's a graphical representation of that data. And so that supported the mechanism of Tipifarnib of inhibiting HRAS membrane localization. We further validated this by using immunofluorescent staining of HRAS. And here we were able to demonstrate that in our untreated sample, HRAS here, which is labeled in green, was localized to the periphery, to the membrane of the cell. When we treated with Tipifarnib, we saw a shift in the HRAS protein to the cytosol and away from the membrane. We then asked if we are able to inhibit HRAS binding to the membrane, would we see a decrease in signaling in pathways that are downstream of RAS? So we performed Western blot on our cells that were treated with Tipifarnib. And we were able to see as we decrease the farnesylated portion of HRAS, we saw a decrease in phosphorylated MEK here in our treated samples, a decrease in phosphorylated ERK, and a decrease in phospho-S6, supporting our hypothesis that we would see a subsequent decrease in signaling downstream of RAS. We looked not only at HRAS mutant cell lines, we compared this finding to cell lines with NRAS, KRAS, and RAS wild type. We hypothesized that this is an HRAS-specific effect. And indeed, we saw when we looked at phosphorylated MEK, we saw a reduction of this protein in response to Tipifarnib in our HRAS mutant cell lines compared to NRAS, KRAS, and wild type, along with phosphorylated S6. We then asked if we see decreased signaling downstream of RAS, will we see a subsequent decrease in cell growth and proliferation? And so we took cells with either HRAS, NRAS, or RAS wild type and looked at cell viability in response to increasing doses of Tipifarnib. So here, highlighted in purple, we were able to see that our HRAS cell lines had a reduction in cell viability in a dose-dependent manner to Tipifarnib. We also wanted to use an additional in vitro model to look at cell growth. So we used soft agar colony formation assays, where we allow the cells to grow in a semi-solid agarose gel that better mimics the way that tumor cells grow in the body. And we were able to again see that in our HRAS cell lines, they failed to form colonies at our higher doses of Tipifarnib. compared to our NRAS cell line models. And so finally, we wanted to look at the effects of Tipifarnib on in vivo growth in RMS xenografts. So we implanted RMS cell lines in mice. After several weeks, we were able to observe palpable tumors. We then randomized our mice to 3 treatment groups to receive vehicle, a low dose of Tipifarnib, or a higher dose of Tipifarnib. We treated mice for 5 days on, 2 days off, which mimics the dosing schedule given to patients. And then we measured tumor volume twice weekly. What we were able to appreciate in our HRAS mutant xenografts, at our highest dose of Tipifarnib, we were able to inhibit tumor growth to varying degrees in this model. This was in contrast to our NRAS and KRAS and wild-type models, which even at our highest dose of Tipifarnib, we were not able to inhibit tumor growth. When we looked at cut and stained sections of tumors at the end of therapy, on H&E, we were able to appreciate a decrease in cells in our highest treatment group in our most sensitive model. And then when we looked at Ki67, a marker of cell proliferation, we were able to see decreased Ki67 staining in our highest treatment group. And then finally, we isolated protein from tumors at the end of therapy to again look at the effects of signaling downstream of RAS. And similar to our in vitro models, we were able to see a reduction of phosphorylated ERK in our highest treatment group and a reduction of phospho-S6, supportive of the results of our in vitro experiments. So we concluded that farnesyltransferase inhibition with Tipifarnib decreases HRAS membrane localization, decreases ERK signaling in HRAS mutant RMS cell lines, decreases in vitro growth in a dose-dependent manner, and decreases tumor growth in HRAS-mutated RMS xenografts. Our findings are supportive of the current study that's open through the Children's Oncology Group, through the MATCH program that was previously described. There's an arm open for patients with solid tumors that are HRAS mutated who can receive Tipifarnib. And we'll continue our work looking at adaptive and acquired responses to Tipifarnib in HRAS mutated fusion-negative rhabdomyosarcoma, trying to identify potential pathways leading to treatment resistance using RNA sequencing and receptor tyrosine kinase arrays. And our hope is to identify additional small molecule inhibitors that might work alongside Tipifarnib to provide more durable anti-tumor responses. And so we're currently investigating combination strategies, looking at other RAS signaling pathway components, including MEK, looking at parallel pathways, including PI3 kinase, and then looking at receptor tyrosine kinases, including HER3. So I'd like to acknowledge all of the members, former and current members of the Pertilis Lab that have supported this research, the members of my advisory committee, Cura Oncology, which has provided scientific oversight to the study, our collaborator at the NCI, Marielle Yo, and then our grant funding sources, including the Children's Cancer Foundation. Thank you.
Are there questions? Yes, Dr. Weeden. While that microphone is going, let me ask you a question as well.
Sure.
This is really exciting because this was considered a dead drug until this work came along, and I think that the persistence of looking for subgroups of diseases where a certain drug might be effective is really important to think about. And since the trial opened in 2020, can you give us a sense of how many HRAS mutant patients have gotten to that point of the match pathway and enrolled?
Yeah, my understanding it's a small handful of patients that we've identified so far. So we're still waiting to see how those patients will do.
And if you look at published mutation data from rhabdo patients with embryonal rhabdo, how many would you expect, what percentage of them would have HRAS mutations of the embryonal group?
Yeah, it's a quite small subset of patients. Looking at data that we saw earlier from Dr. Shern, I believe around 3%, between 3 or 5% of patients in the cohort that were sequenced had HRAS mutations.
So a good case for sequencing patients early so we could possibly find out these things, yes?
Yeah.
Good. Dr. Wiedemann.
Patients, thank you for working on this. As you know, I did several trials, with negative results of Tipifarnib years ago. So for the mouse studies, the exposure or the dose, how does that compare to what we can give to patients? I was just wondering, did you look at a dose that is equivalent to patients?
Yeah, I believe it was equivalent to the doses that were established during those earlier trials.
And then the second question I had, and I forgot, the Tipifarnib, do you have to swallow pills? Because that would eliminate some of the, I guess, young patients that could potentially enroll for the clinical trial.
Yeah, it's an oral medication, I believe, in pill form. I'm not sure if there's a liquid formulation.
Patients, very impressive data. Congrats.
Thank you.
I was most impressed with your mouse curve that you showed in the end as well. And I was wondering if you had, they're really shrink to almost nothing. Did you wait and have them relapse? And can you comment on mechanisms of resistance in those relapses?
Yeah, so we did see that when we followed the mice after we stopped therapy, we started to see tumor regrowth. And so we suspect that the drug works in a cytostatic manner, meaning it inhibits tumor growth but doesn't necessarily kill off the tumor cells. And so that's really what's driving the next phase of our research, trying to identify which pathways are upregulated that are driving this treatment resistance. So that's ongoing work. We've identified perhaps some receptor tyrosine kinase upregulation and perhaps some signaling through non-mutated KRAS and NRAS, but that's work that we're working on right now. So.
Very nice presentation. I was going to ask a similar question. Would you predict that you're going to have to hit two nodes of that pathway, sort of like with the RAF-MEK combined inhibitors?
Yeah, we think so. I think Tipifarnib alone, we're not seeing a lot of apoptosis, so we are looking to see if perhaps we target the PI3 kinase pathway along with RAS. Perhaps we might be able to induce apoptosis. An additional finding that we've seen is some of our cells are differentiating, so becoming kind of more mature cells. And so that's another angle that we're looking at if we can try to promote differentiation using other drugs. And we've seen some cooperation with using trametinib, a MEK inhibitor.
Mm-hmm. Thanks.
I was gonna ask it one more question in a slightly different way. So we know that the vincristine actinomycin Cytoxan combination has been present for actually almost 50 years for rhabdo, and they all would have very different mechanisms of action. And did the Tipifarnib in the trials that were done show much myelosuppression? And if not, perhaps it could be more easily incorporated up front for those patients as a potential synergy early on.
Yeah, I think there was some myelosuppression seen in those trials, some reduction in platelet counts, and we've seen that in one patient that we treated with compassionate use. And so whether or not it could be combined with the VAC regimen, I guess remains to be seen.
Cool. Great. Well, thank you very much.
Thank you.